All right, we are gonna go ahead and get started here, or not. All right, there we go. And we're gonna go ahead and get started. We'll do the last part of chapter 17 here, which means we're gonna be carrying on after today. We'll be done with chapter 17 today. This last portion should take only a few minutes. It's not too cumbersome at all. Some of the stuff is stuff we've mentioned before. We'll go a little bit more detail here into about mutations. Okay, and now we've discussed the past couple of lectures here about transcription and translation, both of which are based on the genetic code, right? And mutations are basically just changes of the genetic code. Okay, and there's many different ways that you can change the genetic code. We've talked about some wholesale changes um, to the genetic code in terms of chromosomes. So you can delete portions of chromosomes, you can duplicate portions, you can invert portions, you can translocate or switch different chromosomes from one to another. But those are big chunks of DNA that get moved around or changed, and the ones that kind of jump up and get you, the ones that a little bit more, I think, sinister or, in my opinion, sometimes amazing, are these things called point mutations, which you see right here. I like the. Let me get the pointer up here. Are these things called point mutations. Okay, and the point mutations are chemical changes in just one base pair of a gene, and sometimes that's all it takes. And the example that I'm going to utilize a lot, and we're not going to get into evolution, but one that I've talked about before is sickle cell anemia, and that's one that I'm going to talk about today as well, a little bit more in depth. Okay, now when you get these point mutations, it causes abnormal protein production. Okay, and these point mutations manifest in a couple of different ways. There's something called a base pair substitution. Okay, base pair substitution is nothing more than you had one letter and you switched it out for another one, or the mutation happened to switch it out for another one that might have been different. Okay, base pair insertions or deletions are the other kind. Base pair insertions or deletions can oftentimes be much more devastating than base pair substitutions, and we'll talk about why as we go through. Okay, so base pair substitution, as I mentioned before, um, replaces one nucleotide and its partner with another pair of nucleotides. So let's say one of the nucleic acids that you had put down had an adenine attached to it. Okay, so it left an A in the genetic code. A mutation that would yield a base pair substitution would take that A and do nothing more than switch it for like a G. Okay, so instead of an A, there's now a G there. Okay, and so what this can do is a couple different things. It can cause a missense mutation, okay, an amino acid, but not necessarily the right one. It can cause there to be an amino acid created, but it's not the right amino acid that should be there. Okay, so that's one way in which these base pair substitutions can really muck things up. Is it creates an amino acid, but it's not the right one, and that's really what we see happen in um, sickle cell anemia. Okay, we also have these ones called nonsense mutations, and nonsense mutations change an amino acid codon into a stop codon. And we talked about what stop codons do, right? Stop codons stop the protein in its tracks. It just stops it. It's as if it gets this false message that says the protein is now done. So oftentimes these nonsense mutations cause the protein to be much shorter than it should be, and if it's much shorter than it should be, it's probably missing a bunch of its structure, which probably means it's not working. So the nonsense mutations can be particularly bad, okay, particularly nasty. Now, what we see up here is examples of both of these things here, okay? We see the original, in this case, the mRNA strand, okay? And the mRNA strand we know has been created from the DNA strand. And down here in these kind of peach boxes, it illustrates what would happen if we have some kind of mutation happen to a single base pair. So the one we're going to look at for this first peach box to compare to the one we see up here is this C right here where the pointer's at. We notice that it changes into a U instead of a C. Okay, and for what we see, that's not really a deal breaker. Okay, that's a mutation that doesn't really matter. It doesn't do anything, and the reason for that is because it happens in the third base pair of a codon. Okay, remember our codons, and we remember that wobble effect that is there because of the tRNA. The fact that that third base pair, if it gets to be wrong, it doesn't really matter. Okay, so in this case, the fact that the C got substituted out for a U just doesn't matter because GGC for a codon codes for glycine the same way that GGU does. So there we see that redundancy of the code that actually saves this protein. Okay, and so that's what we see happening here. We see this protein is now saved. There is a mistake. There is a mistake here, but the protein is the exact same as it would be up here. Okay. Other two are the ones that cause some nasty stuff to happen. Okay, the missense mutation. The missense mutation. We're looking at the same codon. All right. So in this case, we're looking at the GGC codon that codes for glycine, the amino acid glycine. With a missense mutation, instead of this first letter in the codon being a G, let's say it gets switched out for an A instead. Okay, so GGC, if we went to the codon chart, actually codes for glycine. But AGC, if we went to that same codon chart, AGC codes for an amino acid called serine. Okay, and so what that does is it fundamentally changes what we see as the protein. The protein is no longer methionine, lysine, phenylalanine, and glycine, which has a specific structure and a specific function. Now, just because we changed one letter, it changes this last amino acid, and it will probably cause this protein to fold in a completely different fashion and thus have a completely different function as well. Okay, I believe this is the example that we'll see, or a very similar example to what we'll see with sickle cell. I think that's up in the next slide. Down here we see the nonsense mutation. All right, the nonsense mutation causes one of your codons to switch and turn into a stop codon. Right, so we see up here the second codon is where we're looking. AAG is the normal version, and if we look at the second one, this first A gets turned into a U. Okay. Now, if we look at the codon chart, AAG up here codes for lysine. But if we look at the codon chart again, UAG, if we go to that codon chart, says it codes for what's known as a stop codon. So if a stop codon is coded for at that particular spot, then the protein's just going to stop. It will truncate. So this protein that should have been 4 amino acids long is now 1 amino acid long. It got started and then it stopped. Do you think that protein right here, we'll wait for that thing to go away, that protein at the bottom, you think that protein at the bottom is going to have any function whatsoever with 1 amino acid? Absolutely not. Okay, so that's the drastic effect that these things can cause. All right, now I've been harping on this sickle cell anemia thing, right? Well, up here in this picture, we see the normal wild type hemoglobin DNA codes for CTT. Now, the scientists have understood this very, very well. They've narrowed it down to exactly one spot where this mutation occurs. And it happens in that particular triplet code, which codes for this codon. I'm trying to use my fingers, but okay, which codes for this particular codon right here. Normally, mRNA gives you this codon of GAA, and that codon of GAA codes for glutamic acid, GLU. But if we have a single base pair substitution, which is exactly what we have, instead of CTT, now that middle T turns into an A, and that changes this codon to be GUA. Instead of coding for glutamic acid, it codes for valine. Okay, and that's what happens in sickle cell anemia. Now, in the next slide, not this one right here, there is a video. I'm going to link that video in today's assignment, and I would like you to watch it because it talks about the actual talks about the actual structure of the hemoglobin. So what this does is it creates these hemoglobins, right, which are, we know, are proteins, and it folds them in different ways. This this protein hemoglobin is folded in just the right way, and it carries oxygen the exact same way it should. This one right here, because of this one single amino acid difference, gets folded in a really wonky way, and it binds up oxygen a little bit different. And when it does bind to oxygen, it causes your blood cells to like distort and get this really weird shape, which they call a sickle cell. Okay, and that's what we see right here. So this is a normal blood cell, binds oxygen just fine, stays in that kind of lifesaver conformation. But when we see sickle cell, that defunct amino acid, that wrong amino acid, that missense mutation, causes this crazy looking sickle shape. They call it a sickle. It's like a half moon sickle. Causes this thing to change from this normal donut shape to this weird kind of crescent moon shape. And that doesn't seem like it would be a huge deal, but it turns out that it really is a big deal. These things don't work very well when they go through your blood vessels. They clog up. There's big stockpiles here. This they clump up enough that they cause strokes. They cause massive amounts of pain in some people, circulatory issues. It's a really, really bad, debilitating disease, and you see it a lot. There's a lot of people who have sickle cell anemia. Some people die from it, some people don't. It is kind of a graded disease. Some people suffer from it very, very badly. Some people don't, which is an odd way to look at this disease. But chances are somebody who has sickle cell anemia. They have their ways of dealing with it. Sometimes it's bad enough where they have to stay home. Other times they can go to work or go to school. Okay, the title up here says advantages. Obviously, we know there are disadvantages, right? Those are the ones I just listed. But there are actual advantages, and I have touched on these before. And since we're not going to do the unit on evolution, I'm just going to address them very quickly here. Sickle cell anemia is heterozygous. Is a recessively inherited disorder, which means you have to have two recessive alleles to get it, right? If you have no recessive alleles, you don't have it. You have normal blood that looks like this. If you are heterozygous for it, you have blood that looks like this, but you're a carrier of the disease. Now, one thing that you'll hear a lot about, or you'll notice, is that a lot of people who have sickle cell anemia are African American. They have ancestry in Africa. And one of the diseases that's absolutely prevalent and kills a ton of people in Africa is malaria. Okay. Now, it turns out that people in Africa who actually have sickle cell anemia have a very large immunity to malaria. The parasite malaria just can't bind properly to these blood cells. So the people who have sickle cell anemia don't have malaria. On the flip side of that, the parasite that causes malaria can bind really well to these right here. Okay, for people who are, let's say, homozygous dominant for normal blood cells, people like that get malaria like crazy. They come in contact with a mosquito that has it, they're going to get malaria. But there's this middle ground, which I obviously haven't depicted here, but the heterozygotes. There is a heterozygous advantage. You kind of get the best of both worlds. You have normal blood cells, but something in that blood cell causes it to not attract the malaria parasite nearly as much as this with somebody who's homozygous dominant. It's a really fascinating thing. So that's one of the reasons why we still see that recessive trait in the population, because there is some advantage to having it, because you don't die if you have malaria. So, or if you contract malaria, you're not going to die if you have sickle cell, or at least one allele for sickle cell. So you could either die of sickle cell and not die of malaria. You could not die of sickle cell and die of malaria, or you could be lucky enough to be the heterozygote in the middle that doesn't die of either one, just get a little bit sick. I know I'm kind of brushing it off as something simple and funny, but it's not. It's kind of a crazy phenomenon. All right, so I'll link that video. The other types of um, point mutations that we're going to talk about real briefly here are insertions and deletions. Insertions and deletions tend to have really, really just nasty effects, these disastrous effects on people. That here, um, when you insert or delete single nucleotides, it only has to be a single one. What you do is you change the you change the reading frame of what your ribosomes read on the mRNA. If you remember back to, I think, the very beginning of this, I'm not going to flip all the way back, but there was a little graphic with Clifford the big red dog eating a cat. And in the correct reading frame, it says, the big red dog ate the cat, or something of that nature. And then there was another sentence that had shifted the reading frame. Okay, it had caused a frame shift. And what happened was, all we did was we put the spaces in a different spot. And when we put the spaces in a different spot, it created this sentence that made no sense whatsoever. And all we did was shift it by one spot. And that's exactly what happens with these insertions of a single base or deletions of a single base. It causes these frame shift mutations. Everything up until that mutation, that insertion or deletion, is read just fine. But once that frame shift happens, everything after it is screwed up. Absolutely everything is wrong. You get every single amino acid that's in the wrong order. It's the wrong amino acid. It is large scale chaos in the gene if you have everything shifted over by one from what it should normally be read as. Okay. So there's a lot of different diseases. We don't have time to cover all of these. We see some of the common ones: bowel cancer. That's gross, right? Cystic fibrosis, Tay-Sachs disease. A lot of these are just really, really bad diseases. So we're not going to get any more into that, but you you see some of the more disastrous genetic diseases because of insertions and deletions, and all it takes is one base pair, one extra base pair that shouldn't be there, and every gene after that is messed up. Okay, so it's a really, really bad thing. All right, and there's also a video that's linked here that I'm not going to play on this slideshow, but I will link it with the slideshow in Classroom. Okay. So here is another graphic again that shows frame shift mutations. And I'm not going to go through it the same way I did the other one, but here you can see what your normal protein strand would be. In fact, it's the exact same one. And then what we do is we look and we're gonna see, all right, let's put an extra U right where my pointer's at. All right. So we put an extra U in there. Oh, right off the bat, we screw the whole thing up. We don't even get a protein. And everything else after that's probably going to be screwed up. Okay. Here we're going to take out a U. So we're looking at this codon right here. This codon right here. We're going to take out this last U. And that's exactly what they show here. This U is gone. They take it out. And so we get methionine, lysine. Those are perfectly fine. But when we take that U out, instead of reading UUU, we read, and getting phenylalanine, we read UUG, and we get leucine. Okay, the G comes from here. Everything's shifted over one. We took that G out or that U out, everything else gets shifted over. Now, instead of this one reading GGC, we have a different reading frame, right? We now have GCU, which codes for alanine instead of glycine. The whole thing's messed up, and you can imagine here what would happen if you take out a whole codon. Okay? I mean, a whole codon missing doesn't seem like a big deal because you're actually not changing the reading frame here, but you're removing an entire amino acid. That has its own downfalls, right? That's a really bad thing in and of itself. You just don't have a complete protein after that. All right. The things that cause these called mutagens. Spontaneous mutations can occur to DNA during DNA replication or recombination or repair. So mistakes just happen when you're replicating your DNA, when you're making extra cells, when you're making transcripts. Mistakes just happen. That's just a natural thing. You're dealing with 3 billion base pairs. There's going to be a mistake that happens every now and then. Okay, great. But there are certain chemicals that cause mutations to happen more often. Okay, and those are called the mutagens. It could be physical or chemical agents that cause mutations themselves. Okay, so things like UV, that would be an example of a physical type of mutagen, UV rays, they have disastrous effects on your DNA and your cells. And they break the DNA down and they destroy parts of it. And if that DNA goes to replicate, there might be a mistake in it, and every cell after that might be cancerous. Chemical agents like the chemicals found in cigarettes and asbestos and what's the one the weed killer, whatever that weed killer was that got a lot of people non-Hodgkin's lymphoma very recently. I can't remember the name. Roundup. Roundup. Those are chemical agents that chemically interact with the DNA in your cells and then cause chaos. All right. So we're done with chapter 17. Not sure which chapter exactly we're going to go to next because there's some things that we will need in some of the next chapters and some we won't. So I got to cherry pick exactly what we're going to do here. All right. So I will see you soon, and I'm going to get out of here. It's late.